screen?
Yeah. Yes.
Yes. Hi, everyone. Really nice to see that so many people joined today for the Open Lab Day. And I want to present. So my name is Gennady Roshchupkin. So I'm leading Computational Imaging Genomics group in BIGR. We're working on the various topics related to the data integration between imaging and genomics, gene data, gene expression, your mutation, your DNA. And of course, I don't have enough time to cover everything. I want to focus on the projects what we proposed today for the master thesis. But I want to start actually with genomics information, and just to want to make in this presentation clear why it's so interesting to do such type of the analysis. And to start with just some basic background from probably from the high school, that all of us has some in your nucleus you have your double helix structure of your DNA, and this DNA consists of these coding letters, nucleotides, and they encode not only genes but a lot of information which is actually known and unknown. But what is interesting that even between two random people on this call, we have millions of the mutations or variation in these letters, which might be silent, so they don't cause any difference between us, but a lot of them cause differences in like height, BMI, hair color, eye color, but also medical conditions. And that's why it's interesting to study, to understand the cause of various diseases. And in this simple example, in simple analysis, if you just compare two groups of people, for instance, with some disease, what one of the example what was just mentioned during the talks today, let's say dementia or Alzheimer, and then you just compare these letters one by one, and then you can see that here the example, the red one, which is highlighted, the people into groups, they're different in this letter. So it means that this mutation, particular mutation, might be related to a risk for developing Alzheimer's disease. And then you check all these millions of letters, and you can make a picture of this mutation on your DNA, and then you can link it to functions of the genes and what's going on in your cell. So this is actually quite a novel area of research that just recently was 15 years since the first type of such analysis. And on this picture, it's the huge field and a lot of various approaches involved, but the major idea is from the moving from this DNA mutation to understanding of the functionality of this mutation, and potentially to translation to the drug development and clinical practice. Because for the many diseases, like I mentioned, neurodegenerative diseases like Alzheimer, Parkinson, or psychiatric diseases, or also high blood pressure, diabetes, etc., you have genetic predisposition. And there's a huge community. We're also working on some bioinformatical tools to analyze this data, so which I will not cover in my talk today. And also working on the deep learning methods to do such type of analysis. And why you ask imaging is important, because coming back to our study, so that in the example of the two groups of people, we have just case and control. So someone has disease, someone doesn't have. But most of the time, one way or another, you have some underlying continuous measurement, because in clinical practice, you just make a binary decision that you are sick or not sick. But there are, of course, a lot of processes going on in your body. And that's why, as you can see on this picture, at some point, you just cross the line and you become, let's say, a case, or like you, doctor making decision that you're sick. But it would be more important to understand the underlying processes and to extract some more quantitative continuous trait for future analysis, because it can give you more power and more understanding of what's going on. And therefore, let's say, just coming back again to the simple example, then instead of checking the case control study, you can just measure some phenotype, in this case a head size, and then to do the same procedure and see which mutation is responsible for difference in head size. And that was a simple example, but actually coming back to real practice, if we look at one of the most amazing organs in our body, the human brain, that's fascinating and really important to uncover which genetic mutations are important for instance, our cognitive functions or related to the brain volume or specific regions of the brain, again, to link it to diseases such as psychiatric or neurodegenerative diseases. And we are working a lot on the brain genetics, studying structural data with various brain segmentation regions, with the DTI data related to the connectome connectivity information from your human brain, or actually developing a new method to extract different measurements, like in this example, some shapes of the subcortical structures. And it's not only about how you define these regions, but also we can extract the various biomarkers. And Brian mentioned that we, before his internship, was a project about brain age. Basically, we developed a deep learning model to predict brain age based on the just MRI images. And then we showed that if your brain age is deviating from your chronological age, you have a risk factor for developing dementia in future. And as Brian also showed something similar, that you can actually see which part of the brain is important for this prediction. And what Brian was doing, he was trying to actually combine this data with genetics and do some certification, because some genetic factors might also increase your risk for dementia. But it's not only limited to the brain, because brain is one of the examples of imaging data what you can use. Another example that we're working on is facial analysis, because that's the same. For one hand, we're quite similar with our face, but there are a lot of variability, and actually face serves as the health mirror for your global health condition. And because that's also highly genetically defined, you look similar like your parents or grandparents or your brothers and sisters, but also a lot of environment factors and your lifestyle affect the face. And therefore, we're working on the methods how to extract this information from your face morphology, which is also not really simple, because you can't just measure some distances on your face, etc. So therefore, we're working on specific deep learning methods which use some dimensionality reduction techniques, which can reduce your face from thousands of measurements to just some latent features, in this case 200s, and reconstruct it from that 200. And that allows you to analyze these underlying features in relation to the health condition or some, well, in this example, that's just basic characteristics, but you can also link it to the cardiovascular problem or genetics or behavioral problem in kids. And this is something similar what methods I use now in Snapchat or Instagram and so on. So, but in this case, we're applying to the clinical settings. And 3D imaging in the group is not limited only to the face. We're also working with the 3D skull data for kids with the craniosynostosis as well. You heard today about the brain study for the kids with the craniosynostosis, but we are working with the 3D images of the skull, because there's also a biomarker which is important also for potential genetic analysis, but also for surgical planning. And we are working on the deep learning model, which can, for instance, based on your child with the craniosynostosis, to predict the healthy shape of the skull, kind of the gold standard for the clinicians, which they can use as a reference, which gives much more information compared to just opinion, for instance, of the surgeon, how the surgery went. But I mentioned that in genetics, we're also working on the deep learning, but the main problem in genetic application of the deep learning, that the black box concept. And because, as I said, we not only want to predict something from your DNA, but also want to understand why it works. And that's, of course, one of the issues in deep learning community. And there are a lot of methods for explainable AI for image analysis, but we are working on the explainable AI for genetics. And Arno, one of the PhD students in the group, he's heavily involved in the development of such technique on the front line of the basically research. That's quite novel idea how we can integrate biological knowledge for interpretation of the deep learning in genetics. And if to summarize that research directions of the group, that's quantitative trait analysis. So as I mentioned, for instance, brain analysis or 3D image analysis for genetic applications, new method development for omics. That's one part what I mentioned, it's explainable AI. And second part is the distributed machine learning. And also third direction is actually data visualization and accessibility. That's about open source tools for the community, but also for the online tools and visualization tools for other researchers to be able to use our frameworks. And to summarize, I want to mention the projects so that please join our chat room, and Lau, Arn, and Tarek will be there, and they will can tell you more about the projects they proposed to work on. Thank you.